Good morning. Thanks, Stephen. So I'm going to talk to you about the work that we do on multimodal data fusion, specifically for precision medicine, and then also how we plan to use multimodal modeling in digital twins. So we argued that the reason to focus on multimodal modeling is that each data modality for cancer patients has strengths and limitations. Data for cancer patients comes in different forms. There's quantitative features like RNA-seq profiles, mutations. We have lots of images in cancer, digital pathology images. These are high-resolution 2D images. We have 3D radiographic images. And then there's maybe text reports from clinicians, from pathologists, radiologists. So one of the topics that we focus on in the lab is how can we represent these data? How can we learn lower dimensional representations? Because most of these data are very high dimensional. We can't model the raw data directly often. So can we learn lower dimensional representations that then we can combine in fusion models? Also, we need to take into account missing data. There's lots of various types of missing data often. Sometimes we also don't have all multimodal data. And so this is the topic of research in the lab for the last decade or so. So we developed robust methods that can handle various data types, also in the context of missing data. We use concepts of pre-training and transfer learning and foundation models. And then at the end, I will talk a little bit about how we propose to develop multimodal digital twins. So this is sort of how we see the different data streams for cancer patients. So there's electronic health records. This can be labs, notes, history, patient history. Then there's all sorts of molecular data. Routinely collected molecular data is now mutation panels. There's sometimes also RNA-seq. Spatial omics is now becoming an important tool in understanding the spatial biology of tumors. Then we have digital pathology. These are high-resolution 2D images that give us spatial architecture, cellular and cell type. And finally, also radiology, which is non-invasive. We can use this also to track patients over time. We get data about how tumors grow and shrink in the context of treatment. And so the focus of the lab is really to learn these representations. We essentially look at all these different modalities. Then we represent them into vectors, right? These are often nonlinear models that represent the data into vectors, and then we can combine them using all sorts of fusion methods. This is a recent perspective that we published, and we show essentially, or we argue that especially for cancer patients and complex diseases, that using these different, combination of these different modalities can help and make predictions about treatment and prognosis. So today I'm just going to give you a very brief overview of a number of projects that we have done. First I'm going to talk about data imputation, cross-modal modeling for data imputation, also generating synthetic data using RNA sequencing and H&E images. I'm going to show you some of our work in radiology and how we represent lung and brain tumors from radiographic images. Then some examples of multimodal fusion, and then at the end I will talk about digital twins. So first, I'm going to talk about what we call cross-modal modeling, because I already mentioned that we don't always have all modalities for all patients. We often have missing data. Also, some modalities are more routinely acquired than others. Digital pathology and radiology are routinely used to monitor cancer patients, but for example, RNA sequencing is not. Whereas it's important for example, subtyping. We can use it for drug discovery. We can get activity, pathway activities from these profiles. At the same time, an H&E image and whole slide image is the cornerstone of diagnosis in cancer patients. So we asked the question, can we use the image as a surrogate for an RNA-seq profile? So that's what we did. So we developed a model across these cancer tissues. We have as input a whole slide image. Then we have a vision transformer model, which we call Sequoia. And this model essentially predicts the whole RNA-seq profile. We use these cohorts here to develop this model, the Cancer Genome Atlas cohort and also GTAX. This is a cohort of normal tissues and both matched whole slide images and RNA-seq profiles. We had a few cohorts for validation. I'm not going to show the spatial visualization and validation that we did, but we can also, because the model works on patches of the image, we can, even though the model is trained on bulk RNA sequencing, we can still visualize the model spatially. And we also look at which genes we can reliably predict from the image. So this is the model in more detail. So we have these patches. So these are, for example, 250 by 250 patches that we grab from the image. Then we use a recently developed foundation model that is essentially a feature extractor. So what it does, it reduces this image into a feature vector of, let's say, 500 features. Then we have a clustering step, and this is really to capture the heterogeneity in the image. There's lots of different cell types in the image. And this gets then fed into either of these modules, for example, a transformer that then predicts gene expression. So let me show you some results. So we look at the correlation coefficient between the predicted genes and the true expression, and we also look at how many genes we can reliably predict from the image. And we focus on high correlation, but also low mean square error. And then at the top here, I'm comparing these three different ways of making the final prediction. Here is the result for breast cancer. You can see we have high correlations of 0.6 and higher, and we can predict up to 17,000 genes with high correlation and low error. Also, the uni model is significantly better. So these digital pathology foundation models are significantly better than previously developed models on ImageNet. These models that are trained on specific images, medical images, do now, since a year or two, outperform these models. And also the linearized transformer also had the best result, and this is across cancers. And you can see that across many different tumor types, we can predict thousands of genes reliably. And this is true for also a number of other cancers, for example, lung cancers. So this first example, so it's essentially we developed this vision transformer to make a prediction of gene expression. I didn't show you this, but we can also spatially visualize the prediction of the genes, and we show in spatial transcriptomics data that it correlates well for certain genes in glioblastoma. So now we also thought, can we do the opposite? Can we actually use an RNA-seq profile and generate a synthetic image? And this is, again, motivated by for the same reasons. And another reason is also to generate synthetic data and to have advantages in terms of data sharing. So can we generate synthetic images that then we can share more easily? And also, can we generate synthetic data for diseases that are less prevalent? For example, pediatric cancers. Can we oversample them, essentially? So in the previous talk, this was also shown, right? There's these models that are very popular in terms of generating images from text. And essentially, our model is modeled after that. So we treat an RNA-seq profile as text. Then we have a cascaded diffusion model that is conditioned on this vector and then generates an image. And this is motivated by the success of previous models in this area. We previously looked also at generative adversarial neural networks, but these have some drawbacks because we want a single architecture to model across tissues, whereas with GANs we were not able to do that. So we initially trained to model for 5 tissues, and now we have updated it for pan-cancer. And then we show that synthetic images that we generate essentially capture the same cell types that we see in real tissues. And then we also use these synthetic data to train new models, and we show that we can replace, essentially, partially replace real data. So this is the initial model that we trained on 5 tissues. These are the tissues that we used. So this is the model. First, we needed to represent the RNA-seq profile. So we essentially have a variational autoencoder that reduces the dimensionality to about 200 features. And this is a variational autoencoder, so we learn means and variances. And then once we have this embedding, we have a cascaded diffusion model that essentially is conditioned on this embedding and then iteratively learns these images. We first learn 64 by 64 images, and then we upsample this in successive steps. And these are some results. So you can see glioblastoma and lung cancer, synthetic images at the top and at the bottom. we showed these two pathologists, and they cannot distinguish real from synthetic images. This is another visualization of the 5 tissue types. In the middle, I'm showing you the RNA-seq, a UMAP of the RNA-seq profile. As we know, RNA-seq is very tissue-specific, so we see nice clusters for each of the tissues, and then very specific, tissue-specific, let's say, visualizations in these synthetic images. And here is one example of how we try to show the fidelity of these images, of these synthetic data, and that are useful. So we use HooverNet to detect cells and to classify them in a number of different cell types. And then we see this is for lung adenocarcinoma, that in both the synthetic and the real images, we see the similar cell type profiles. And the same here for renal cell carcinomas. Then we also want to use these data, synthetic data, to pre-train models. So we can train models on synthetic data and then fine-tune them on real data. And we see when we add synthetic data that we have a better performance, for example, here in predicting a macrosatellite instability in colorectal cancer. And that's true for both accuracy and F1 score. So now we have extended this model. We have now a pan-cancer model, and we can generate images across all tumor types. And we are now working on trying to prove that if we generate, so we have generated now 20 million images, and we are developing foundation models on mixtures of real and synthetic data, and figuring out where does the model break down, right? How can we achieve similar benchmarking performance with models that are trained on synthetic data versus real data. So now let me show you some work that we have done in radiographic images. So in this context, what we are doing is we're essentially doing unsupervised learning or self-supervised learning. So this is very similar to what I mentioned in the beginning. We want to learn these lower dimensional representations of 3D radiographic images. And essentially what we do is we use these autoencoders, 3D autoencoders, to represent lung and brain images. So here is the workflow. So we have these 3D images. Then when we essentially represent them using an autoencoder, then we have an embedding, a lower dimensional feature vector that then we can use for predicting all sorts of downstream tasks, including treatment response. And this is a general approach. We can use this across different types of organs. So we trained a model like this on various lung cancer cohorts with lung CT imaging data. And the main thing I want to show you is that we can, so the model actually captures tumor size in these images. So here I'm showing you a number of Stanford cases with average size, lung tumors. And we essentially used a difference factor of the smallest and largest lesions. And then what we can do is we can actually grow and shrink these lesions. So what we want to do now going forward is we want to train these models on longitudinal profiles of lung cancer patients and capture tumor growth trajectories of lung cancer patients. Similarly, for brain MRI, we've also developed similar models on multimodal MRI. We used a number of different cohorts. We used brain MRI from UK Biobank, from healthy individuals. And then we test the encoding that we essentially learn on various other cohorts, disease cohorts, both in oncology and also outside of oncology. And these are the architectures that we use. These are all various types of autoencoders. We have UNETs, a regular autoencoder, variational autoencoder, and so on. And essentially what we observed here is that in terms of reconstructing the image, a UNET is always the best. But a UNET does have these skip connections, so essentially cheating in terms of reconstructing the image. But it doesn't actually incorporate a lot of information in the embedding factor, which is what we want. So in terms of correlating the image, the essentially unsupervised representation of the image with phenotypes, for example, in UK Biobank, the regular autoencoder had the best performance. And similarly, then we wanted to show how we can use these models into predicting overall survival in glioblastoma. And then again, we see the regular autoencoder in 3 cohorts, TCJ, UCSF, and UPenn. We see a nice prediction of overall survival in glioblastoma patients. This is just an example of how we essentially want to model radiographic images using these autoencoders, and that essentially gives us a feature vector that then we can correlate with downstream tasks. So now let me show you a few examples of multimodal data. So essentially this is our vision in terms of modeling multimodal patients. We have, again, these four modalities that I mentioned in the introduction, but often some of these modalities are not present. So then we can use some of these cross-modal models to impute the missing data. And this is not unusual. Missing value data imputation has been a common research topic in the past. And what I've shown you is some of this cross-modal modeling. And essentially, we propose to evolve into this idea of synthetic multimodal model, where we can generate also multimodal data of new patients. And let me show you a few examples. So here are a few examples of multimodal models that we have done in the past. So we have previously developed a multimodal framework for multi-omics and whole slide image data that were encoded into feature vectors and then used to predict survival. And we showed that in many cancers that improved prediction of prognosis. More recently, we've also done multimodal modeling in the context of immunotherapy response prediction, where we essentially combine whole slide images with RNA sequencing and spatial proteomics data, showing that when you combine features that predict immunotherapy response across all these different modalities, we had essentially the best result in terms of predicting outcome of immunotherapy response. And also in the context of brain tumors, adult and pediatric brain tumors, we've developed a framework to combine RNA sequencing and digital pathology data, and essentially showing that fusion of these different modalities results in a better performance for predicting prognosis. And also we did some work in transfer learning between pediatric glioma and more rare pediatric tumor types such as ependymoma and medulloblastoma. So essentially, to summarize, so the work we've been doing in the lab is really focused on this idea of multimodal modeling, taking advantage of recent advances in building foundation models on unlabeled data. And then once we have these feature extractors for each of these modalities, then we only need to learn sort of a fusion layer that combines across multimodal data. And current work in the field is really focused on this next step. So now, how does this connect to a digital twin? So we recently proposed how mathematical modeling can connect with deep learning to essentially develop multimodal digital twins. And the idea is then to have this concept of an in silico patient that we've been talking about today and yesterday. And we think that the combination of mechanistic modeling and multimodal foundation models is the path forward. Because using these multimodal foundation models, we can generate synthetic data, also potentially longitudinally, that then can be used to initialize mathematical models. Let me summarize, and these are the people that did the work and my funding, and thank you for your attention. Happy to answer any questions.
Thank you very much. We have time for a couple of questions. Yes, Trevor.
Seeing the RNA-seq is very impressive. Is it some genes are just clearly like breast cancer genes and other genes are clearly colon cancer genes? Is that what it's seeing? Or is it actually seeing something specific about individual tumors?
So it is tissue specific, right? You saw that within each cancer tissue.
Tissue specificity, was there more the tumor specificity that was interesting?
So there is, yeah, I can't, off the top of my head, comment on sort of what genes overlap across all the tissues. But I would say that there's definitely some tissue specificity. As you can see, as you saw, that for a breast cancer, we had the best result. For glioblastoma, it was the worst result. I think some of that was due to the dataset size. So we see a correlation between how much data we have for each cancer tissue. I think also in terms of like what genes we can predict, I think we also need to consider that not all gene expression is independent, right? There's high correlation, so it's not that surprising that we can predict so high number of genes, because the number of sort of independent random variables is likely limited in a RNA-seq experiment. Not sure if that answers your question.
I mean, things like if you had 100 tumours with a H&E image and you had 100 RNA-seqs, is the prediction of the 100 from the 100 H&Es equivalent to the heterogeneity you would expect from the 100 RNA-seqs? But does the variance match as well as the mean? I can see you can predict the mean quite well, but... Sure.
Well, I do have to emphasize that this is bulk RNA sequencing, so we're getting RNA sequencing of a neighboring sample, right? That's not necessarily exactly the slide that the image represents. So it is sort of an association, right, that we're learning, yeah.
Just interested to hear your speculation on how the same model Trevor was referring to kind of deals with the well-documented thing about transcript levels not correlating with protein levels or metabolite levels or sugar levels. So, how does it cope with that lack of correlation? What's going on under the bonnet?
Unfortunately, yeah, there's very few protein level or let's say whole protein level data sets out there, so we haven't been able to study that in detail, especially in terms of like the link between imaging, like whole site images, and proteome. So yeah, I think that's a hard question, and it's very costly to generate proteomic data.
Yeah,
so
I'm not saying that you need to have kind of spatial proteome data. It's just obviously when you're looking at the H&E images, that in some way is integrating what different types of proteins they have, their specificity, their acidity, all of these sorts of things, and it's giving you an integration of all of that. But if that which is staining proteins mainly, and the proteins themselves are not that correlated with the RNA,
yes,
how is the problem getting solved? Is it learning kind of somehow the rules of what doesn't correlate and how you can infer from other genes actually that this transcript isn't going to correlate in this context, but it will in a different context? Or...
I think the main thing that we're learning is cell type, right? So, and there's RNA products that represent cell type, and there's also protein products that are representing cell type, right? So what we're learning in the image that predicts the expression is most likely is the cell type and the structure, and so that's probably what's mostly driving that prediction. But there's lots of factors that translate RNA to protein, like alternative splicing and post-translational modification, that make that relationship very complicated, and that is still sort of understudied in a way. Yeah.
I think Sandy.
Well, you can say no to me.
No. Oh, wow.
That would be the fifth time.
It's personal. I think I just want to be very clear.
I'm kidding. But I always have a question, just comes to me at the end, right, based on the discussion. Really intriguing work, right? And part of me can't help but think it's all science fiction, but that's just the mathematical modeler in me. But obviously these are useful tools, and I guess from a modeling point of view, one of the things that I'm really intrigued about is temporal dynamics, right?
Yeah.
So spatial dynamics and dynamics being the key word, and so is there an opportunity here to leverage these approaches to help drive the temporal dynamics? Because one of our big problems clinically is sparse data when you get a biopsy at the beginning and maybe a biopsy at the end, or you get imaging every 4 months or 5 months. Is there a way to sort of correlate in between somehow with these approaches and almost like forecast what's going to happen ahead of time? Could you actually use it for that approach?
So we are now attempting to look at longitudinal imaging data, CT and MRI. We have a few cohorts, lung cancer and brain tumor cohorts, where we have a few hundred cases which we are curating to, and we have about imaging every 2-3 months. And the assumption is that across all these profiles, we would be able to discern some sort of average growth trajectory that would then allow to generate synthetic images in between and maybe sample at higher frequencies. So that's something that we're...
An opportunity there would be actually to integrate modeling.
Yes,
to
yes,
help
of course, yeah.
With
Yeah.
that, you know,
Yeah.
projection.
I think that's exactly what we're trying to do, is to have one feed of the other, right? So we build a model, we see what kind of, because essentially the first phenotype we want to extract from that is just tumor volume, right? We can of course look at the image, but we would just extract tumor volume from that and just look how well that matches maybe a mathematical model and of course the truth, yeah.
So with that, I'll draw the session to a close. We have lunch and we'll be back here at 1:30. So there should be lunch outside. So thanks everyone for this session.